speaking for this audience. I think this Myeloid network is the best network there is. I'm really enjoying it, and it's so nice to be able to follow it also from Europe with all the time differences. And I think it's the best crowd ever. So I'm really looking forward to any feedback you may have. The story I'm going to talk about is really a team effort where we really made a move and connected with the clinic. Many people involved. I will be mentioning those. And it just came out in Cancer Cell as mentioned, but I also incorporated a few new data in there. So also there, I hope maybe to get some feedback from you. So these are my disclosures. So my lab is very much interested in the crosstalk between breast cancer and the immune system. And in particular, we are interested in also the systemic effects of a primary tumor and how this may shape metastatic disease, but also a therapy response. So as you all know, the tumor microenvironment, of course, is very much evolving during tumor progression. There's recruitment of all kinds of immune cells, but we know that primary tumors also affect the immune system at a systemic level, so beyond the tumor microenvironment. Mediators secreted by cancer cells or stromal cells present in tumors, immune cells, they via the bloodstream enter, for instance, the bone marrow, where they modulate hematopoiesis. They often we see accelerated granulopoiesis, premature release of immature myeloid cells. This changes the systemic immune landscape and can also affect pre-metastatic niches, as we know quite well from many studies coming out of people here that are part of this Myeloid network. So my lab also has become interested in well, this interpatient heterogeneity in the immune landscape. So if we look at the systemic inflammation, but also at intratumoral immune cells, we see that even within seemingly homogeneous patient groups, for instance, one particular breast cancer subtype and stage, there's still remarkable variety in the composition and the activation status of immune cells. And here we've been mainly focusing on how cancer cell intrinsic mechanisms shape the way how cancer cells communicate with the immune system. In the lab, we've mainly been interested in genetic aberrations, but from literature, we also know that epigenetic modifications and resulting downstream aberrant intracellular signaling and metabolism, they all change the secretome of cancer cells and surface expression molecules. And this has consequences for how cancer cells communicate with the immune system. So these are processes that we really like to study, because we feel that if we understand how cancer cells manipulate the immune system and how cancer cell intrinsic processes are involved, that this may help us to slowly move towards more personalized immune intervention strategies. Well, we became very interested in how neutrophils are involved in this systemic inflammation. So we, but also many others, have shown that primary tumors can induce neutrophil mobilization and activation. We showed that in particular, when memory tumors lose p53, then we see activation of this IL-1 beta, IL-17 neutrophil axis, resulting in the activation of neutrophils that are highly immunosuppressive and that help disseminating cancer cells to escape from immune attack. So when we identified this cascade in 2015, it was also the year when I was being evaluated as a junior group leader to become a senior group leader at the NKI. And during this process, one of the, I would say, the best advice that I got from the evaluation committee, who luckily evaluated positive about my position, but was that they advised me to make a connection to the clinic. And yeah, I really took this advice serious, and over the last, well, let's say 7 or 8 years, we have been invested in connecting our lab with the clinic. So I still consider my lab as being a fundamental and preclinical research lab, but by having bridged, made a connection to the clinic, I think it really has enriched our research over the last years. And I would like to show during this the coming 45 minutes or so how we made this step and how it helped us also in our fundamental research projects in the lab. So when I got it as advice, well, where do you start? And I think it always starts with a very good partner in the clinic. And at the NKI, we have, Marleen Kok. She is a medical oncologist, but also scientist. And she is spearheading the immunotherapy clinical trials at our institute, focused on breast cancer. So about 7 or 6 years ago, she came back from a research stay in the US. And she started her clinical career here at the NKI, but also having a research lab. And we started to collaborate because we had shared interest in breast cancer and the immune system. And I think this was quite a major investment that we did. She was treating breast cancer patients, and we started together with her team to set up a immune profiling pipeline to start to comprehensively profile all the patients that she was going to enter into clinical trials, but with a special interest to look at granulocytes and myeloid cells. And as you may know, we cannot study these cells in PBMCs because these cells don't survive a freezing step. So we had to do this from fresh patient samples, set up this pipeline. And also here it's important if you, with the circadian rhythm in mind, which really influences in particular neutrophils, we luckily were able to set up this pipeline together with the clinic, so the patients would come in at a certain time, so we would have timed samples from both healthy donors and patients. And just to give you an example, here we looked in stage 4 triple negative breast cancer patients, where we used this flow-based immune profiling platform consisting of more than 50 different antibodies to analyze these, the systemic immune landscape of patients. Just to give you a very small snapshot of the data we get, because of course we're this way generating big data sets. But what for us was quite eye-opening to see is that it's very important to also keep track of the treatment history of patients. So we look here, for instance, at neutrophils, where we see, as we see in our mouse models, really increased levels of neutrophils, but only we see this in the triple negative breast cancer patients that were chemotherapy naive. Patients that were treated more than 1 year ago with chemotherapy, or patients that were recently treated with chemotherapy, we see much a different pattern in their systemic immune landscape. So this way we are trying, we are mapping not only for triple negative breast cancer patients, but for different breast cancer subtypes, the systemic immune landscape. And this sets the basis for, the story that I would like to talk today about. So what happens if we now enroll these patients in immunotherapy clinical trials? So we're not only profiling the systemic immune landscape, we also perform more in-depth analysis, such as proteomics on neutrophils that we can flow sort from blood of these patients. And we see very different patterns, maybe not so surprisingly. But this provides us new research questions that we can then test in the lab. For instance, what we also observed in these patients, neutrophils, that when patients with more advanced disease, we see more ROS production, and this fits with what we observed in our mouse models, that INOS levels in neutrophils are very important for this immunosuppressive capacity. So by mapping the baseline immune profile of the different subsets of breast cancer patients, we quite quickly became interested in understanding, can we use this mapping of the systemic immune landscape also in the context of immune checkpoint blockade? We know that for breast cancer, it's very difficult to treat patients successfully with immune checkpoint blockade. About 5 to 10% of unselected breast cancer patients do show a response. This can be increased when we select triple negative breast cancer patients, also when we select patients that express PD-L1 on their tumors, but the majority of these patients regretfully do not respond to immune checkpoint blockade. So about 6 to 7 years ago, when we started the first clinical trials here with breast cancer patients and immune checkpoint blockade, we became interested in studying this in more depth. So most of the studies on immune checkpoint blockade quite logically focus on the T cells, but as we all know from effective immune responses, that this crosstalk between the innate and the adaptive immune system is really key to develop very effective T cell responses. But the role of myeloid cells in anti-tumor immune responses in this context of breast cancer and immunotherapy is less clear. So here, together with this very talented team, spearheaded by both Marleen Kok and me. So we have Leonie Voorwerk, she's Ph.D. student from Marleen, and she was treating breast cancer patients with immune checkpoint blockade. Hannah Garner, postdoc in my lab, who bridged the clinic and fundamental research by setting up these immune monitoring pipelines. And then Olga Blomberg and Lorenzo Spagnolo, we did do fundamental research in transgenic mouse models. We started out on this project. So what we did is we in parallel did this in-depth profiling of breast cancer patients entering immunotherapy clinical trials, as well as studies in genetically engineered mouse models for breast cancer, whereby we mimic, in some sense, the treatments that we were giving to our breast cancer patients also in mouse models. And then we perform parallel profiling of the mouse and patients, and then use the mouse models to perform mechanistic insights into observations that we did. So the basis, and I would like to start telling this project from the patient perspective, and then move towards more the fundamental mouse model studies. So the basis was the TONIC trial, which is a clinical trial for stage 4 metastatic breast cancer patients that have triple negative breast cancer, and spearheaded by Marleen Kok, where we had access to longitudinal blood samples, but also longitudinal biopsies of the same metastatic lesion. So in this trial, patients were treated first with a 2-week induction treatment, low-dose chemotherapy or low-dose radiotherapy. And for the project I'm going to talk about, this induction treatment is not so relevant because we did not observe differences between the type of induction treatment and the observations we did. But then we also have on treatment blood and biopsy samples. Well, in this trial, about 24% of the patients are responders, either complete or partial responders or stable disease. And as I mentioned, we were able to perform these profiling studies on samples from these patients. Well, just to give you a little bit of flavor of the type of data that we collected. So this is a heat map showing the flow cytometry data that we obtained in all the different immune cell populations that we analyzed by flow cytometry. And well, as you may appreciate, there's every column is a different patient. There's quite a drastic interpatient heterogeneity in the immune composition in blood in these mice. And we were not able, for instance, this is baseline analysis, we were not able to find patterns that would distinguish responders from non-responders. Not at baseline, but also not at the different on-treatment samples. So computational biologists in my lab started then to look at kinetics of immune cell populations while these patients were being treated. And then we started to see well, quite interesting differences. What we observed is that, well, 3 immune cell populations were behaving differently in responding patients versus non-responding patients. And for today, I really would like to highlight the eosinophils that were popping up out of these analyses. So eosinophils at that time, when we identified them, were very, basically overlooked immune cells in the cancer field. There was some literature on these cells, and well, these data were opposing. So some studies would suggest that these eosinophils may have pro-tumorigenic functions in the context of cancer, support tumor growth, shape the pro-tumoral tumor microenvironment. But there are also some studies that suggested that eosinophils may have anti-tumorigenic functions, direct cytotoxicity activity, but also a link with CD8 positive T cell recruitment and activation. And that's, of course, interesting if you think about the context of immune checkpoint blockade. So we had a closer look at these eosinophil dynamics in our breast cancer patients, the responders versus the non-responders. And what we see indeed that these eosinophils were going up in the patients that were responding to immune checkpoint blockade. And we did not see this consistent increase in eosinophils in the non-responders. And we also observed that the patients that had a more than twofold increase in eosinophils during immune checkpoint blockade, that they had an improved overall survival. So it was not so much the baseline levels of the eosinophils, but it was the kinetics of the eosinophils that was connected with response. Well, this is what we measured in blood, but we were also interested to know what would happen inside the metastatic lesions of these patients. And here we benefited from the longitudinal biopsies that were taken from the same metastatic lesion during treatment. So we performed RNA sequencing on these biopsies, and bioinformatician Olga Iseva designed an eosinophil gene signature, basically as a way to get an estimate of the presence of eosinophils in these metastatic lesions. And again, what we also see in these lesions is that based on the RNA signature, that in the responders, there was an increase in eosinophil signature that we did not observe in the non-responders. And we saw that this eosinophil signature positively correlated with a signature for CD8 positive T cells, which we do not see in non-responding patients. So here I show the CD8 T cell signature, but we also looked at active and a signature associated with activated T cells. And there we also saw this positive correlation. Now, while we were picking up these eosinophils in our studies, also other groups started to pick up eosinophils in their patient analysis. So this is an overview, very nice, from the Munitz lab from Tel Aviv. They're experts into eosinophils in cancer context. And here you see a summary of the different clinical trials that have been performed, where they have analyzed eosinophils. And basically in all of these studies, they see that eosinophils are positively correlated with a response for immune checkpoint blockade. Some studies suggest that it's the baseline levels of eosinophils that are positively correlated, and other studies also show, like we do, this increase during treatment. I think what's interesting to see is that it's not limited to one particular type of immune checkpoint blockade, but both the ipilimumab, so anti-CTLA-4, as well as the PD-1 and PD-L1 blocking antibodies, they have seen this. So now, of course, the big question is are these eosinophils just a bystander effect of well, this immune checkpoint blockade, or are they really causally involved in immune checkpoint response? Because if we want to therapeutically use this information, it's very important to know whether they're causally involved. So here, well, our expertise with mouse models came in very handy. So in the lab, we make use of transgenic mouse models for breast cancer. This is one of our favorite models that we use, but we also have other models. Breast cancer is driven by loss of E-cadherin and loss of p53. It takes about 6 to 8 months before these mice have a palpable memory tumor. And then we can start treating these mice, for instance, with immune checkpoint blockade. And here we use a combination of anti-PD-1 and anti-CTLA-4. And as we see in the majority of breast cancer patients, these mice do not respond to immune checkpoint blockade. So not here in terms of survival, but also not based on histology or other readouts. However, if we combine immune checkpoint blockade now with chemotherapy, so this is also in patients, in triple negative breast cancer patients, that see a benefit of combining chemotherapy with immune checkpoint blockade, we see the same in our mouse model. In particular with cisplatin, we see that cisplatin alone already extends survival, but now we see synergy when we combine cisplatin with immune checkpoint blockade. And this is a CD8 positive T cell dependent effect, because if we deplete CD8 T cells, we lose this added benefit of immune checkpoint blockade. So also in this quite lowly immunogenic spontaneous breast cancer model, where we know there's a lot of immunosuppression going on, we are able to well, to push the system, use chemotherapy to obtain immune checkpoint blockade benefit. So like we did in patients, also in these mice, we started to in-depth profile the immune landscape. Well, these mice were being treated with immune checkpoint blockade. First, we looked in blood. So here you see the different immune cell populations we analyzed by flow cytometry. And I think what was very striking, and we obtained these results really in parallel with the mouse, with the human data, is that the only population that systemically and consistently increases when we treat mice with immune checkpoint blockade, but more extensively when we treat them with the combination of chemotherapy and immune checkpoint blockade, are eosinophils. So very much in line with our clinical data. And when we analyze memory tumors, so the tumors in mice, again, we see that in the eosinophils increase. And I think what's important to keep in mind also for the rest of the talk is that systemically, immune checkpoint blockade alone already increases eosinophils a bit. And this is increased by the combination treatments. But inside the tumors, the eosinophils are only coming inside the tumors when we combine cisplatin with immune checkpoint blockade. So very much in line with our clinical data. Well, dearly we looked at eosinophil presence, but what about their phenotype? So there's not so much known about eosinophil subsets, but here we can really benefit from studies done in the asthma field and in the parasite field, where eosinophils are well, very important cell types. And especially in this study where they analyzed asthmatic mice, they identified two different types of eosinophils: tissue resident or regulatory eosinophils and inflammatory eosinophils, based on in particular cyclic F expression levels, but also CD62 ligand and CD101. And well, here you see how they distinguish between these two different eosinophil populations. So we were very interested to study this also in the context of cancer, and also how therapy would change this. So what we show here, this is the cyclic F expression, and what you can see is that these are tumor-free mice. These are untreated mice with a tumor. Here we treat with immune checkpoint blockades, and here with chemotherapy. But you can see that if we treat them with cisplatin and immunotherapy, we start to see the cyclic F high eosinophil population, also quantified here. So this is in the circulation. We kind of see similar overlap with this asthma paper in terms of CD62 ligand and CD101 staining. So what about intratumorally? Do we see the same shift in this inflammatory type of eosinophils? Well, if we look inside the tumors, it's a little bit different. We do not see this division in cyclic F high and low, but what we do see is that already in mice with a tumor that are not being treated, that a large proportion of the eosinophils have this inflammatory type of phenotype, which is not very much changed if we treat these mice. So at this moment, we are very much interested in diving deeper into this, well, potential different subsets of eosinophils and how they are being affected by the treatment. We also performed RNA sequencing on the eosinophils to try to understand them a bit better. I have to say, having worked previously with neutrophils, which are already quite challenging cells in terms of RNA sequencing, I think eosinophils are even worse. There are not so many eosinophils, and the levels of RNA we can get out of these cells is very limited because they are loaded with RNases. So I can only show you a control antibody treated group and the combination treated group where we isolated the eosinophils from the circulation. Where we do see that in particular there's an interferon gamma phenotype in these eosinophils when we treat these mice with cisplatin plus immune checkpoint blockades, and we see other pathways coming up. But also this is something we would like to pursue further, and at this moment we're performing proteomic studies where we have better yields also of all the control groups. But of course, well, the biggest question now is, are these eosinophils functionally involved in therapy response, because we see them going up. We see that their phenotype is being altered. Based on RNA sequencing, we see that they have, well, some kind of an activated phenotype. But are they relevant in this setting? So it's possible to deplete eosinophils using an antibody against cyclic F. So cyclic F is quite specifically expressed on eosinophils. There are also alveolar macrophages that express cyclic F. However, the expression levels are much lower, and we don't have indications that we're depleting other cells besides eosinophils when we treat mice with cyclic F. So when we treat them with this depleting antibody, indeed, we see that the eosinophils are almost completely reduced when we treat with cisplatin plus eosinophil depletion, but also in combination treat with immune checkpoint blockade. And if we now treat tumor-bearing mice, again, with cisplatin plus immune checkpoint blockade, we see therapeutic benefit. But if we deplete eosinophils, we see that we lose this therapeutic benefit. And very importantly, if we deplete eosinophils when we treat mice with cisplatin alone, we don't see that the chemotherapy response is further reduced. So it's really the immunotherapy benefit that we lose when we deplete these eosinophils. Well, we did these studies in a primary setting, but the patient cohort that we analyzed was in the metastatic breast cancer setting. So we were interested to see whether we could see similar observations when we analyze mice with metastatic breast cancer. So we have a mouse model to study this. This is based on the transgenic mouse model that we have. What we can do is we can orthotopically transplant small tumor fragments from these mice into wild-type FEB mice. Then once these mice have developed a full-blown tumor, we can surgically remove these tumors. And then about 2 weeks later, when we do CT scanning, these mice start to develop metastatic disease, mainly in lungs and in lymph nodes, sometimes also in other organs. So what we did is we waited until these mice had metastatic disease, and that's when we enrolled them into immune checkpoint blockade treatment groups. Now, also in this metastatic setting, monotherapy with immune checkpoint blockade, so anti-PD-1 and anti-CTLA-4, does not extend survival at all. Chemotherapy, so cisplatin in an adjuvant setting, does slightly delay metastasis outgrowth or metastasis-specific survival. But when we combine this with immune checkpoint blockade, we now even have mice that are durable responders and that don't respond, that don't develop any metastasis at all. Also in this setting, we observed that the eosinophils were going up in these treatment groups. And when we deplete eosinophils, we completely lost these durable responders. And again, here, this is the immune checkpoint blockade benefit that was gone when we depleted eosinophils. So really telling us that these eosinophils are critical for immune checkpoint blockade therapeutic benefit in our mouse models. So of course, this opens a lot of different new questions. So how then do these eosinophils contribute to tumor destruction when we treat with immune checkpoint blockade? And what are the mechanisms how the treatment is mobilizing these eosinophils and activating these? Because if we would understand these at the molecular level, well, it may really help us to find ways to therapeutically engage eosinophils in the context of immune checkpoint blockade, perhaps as a means to improve immunotherapy for breast cancer. So I will start with the second question. So what do the eosinophils do? So we know from our mouse models that in the end, it are the T cells that are the effector cells. So we analyzed whether eosinophils are connected to the CD8 positive T cells. So first we looked at the abundance of T cells inside the tumors in the different treatment groups. And I think it's mainly relevant to compare the treatment group where we treated with cisplatin and immune checkpoint blockade, so where we have a therapeutic benefit, and what happens when we deplete the eosinophils. So we did not see an effect on the total presence of T cells inside tumors when we deplete eosinophils. But what we do see is that the combination treatment gives quite good T cell activation inside tumors. But if we deplete eosinophils, we lose the presence of these activated T cells. Also shown here in the heat map, where we looked at all the different T cell activation markers. Absence of eosinophils, we lost the activated T cells. Well, how do eosinophils then connect to the T cells? Based on literature and very nicely recently reviewed again by the group of Ariel Munitz, from literature, there are different connections between eosinophils and T cells. Eosinophils can potentially present antigens themselves. They express immune checkpoint molecules. They can secrete ionos, like we observed in the neutrophils, to inhibit T cells. But they're also powerful producers of, for instance, chemoattractants for T cells. So, and they're known to normalize vasculature that may also help to get more T cells inside tumors. We did look at the vasculature in our tumors, and we, based on CD31 staining, perhaps a little bit superficial analysis, but we did not see very large differences in the vasculature in treated mice in the presence or absence of eosinophils. But what we did observe, if we do an array for T cell recruitment chemokines on whole tumors that are from our mouse models that are treated with the combination treatments, cisplatin plus immunotherapy, or when we depleted eosinophils, we do see that one of the chemokines, CXCL9, is strongly reduced when we deplete eosinophils. And when we now treat tumor-bearing mice with chemotherapy plus immune checkpoint blockades, that's when we do see these higher frequency of activated T cells inside tumors. If we now also at the same time inhibit CXCL9, we lose the presence of these activated T cells in the tumors. So suggesting that CXCL9 plays an important role here. Formally, we, of course, with these experiments do not prove that it are the eosinophils themselves that produce the CXCL9. So we hope that the ongoing proteomics experiments that we're at this moment doing, that they may provide us more insights into whether the eosinophils are the direct source of CXCL9 or whether it's an indirect mechanism by which the eosinophils contribute to CXCL9. But this is ongoing, and well, hope to be able to provide you more information at a later stage. So then we decided to study how then does the treatment lead to these systemic eosinophils? Because if we would understand that, maybe we can use that knowledge to mobilize these cells in the patients that are not responding. Now, like all myeloid cells, eosinophils develop in the bone marrow. So we decided to start with the source and look in the bone marrow and analyze whether eosinophil frequencies in our progenitor cells are already increased. And I showed you before that in the mice, the immune checkpoint blockade alone is sufficient to increase the eosinophils in the circulation. So that's why for now I leave out the cisplatin element, but it comes back later. So when we treat tumor-bearing mice with immune checkpoint blockade, we see that already in the bone marrow, there are more eosinophils, and also progenitor cells are increased in the bone marrow, suggesting that the treatment with immune checkpoint blockade systemically increases the eosinophil production in the bone marrow. So we hypothesize that if we now analyze serum from these treated mice, we should be able to pick up the mediators that drive this accumulation of eosinophils. So that's what we did. We treated mice with immune checkpoint blockade or with a control antibody, and we performed a big array for eosinophil-related cytokines and chemokines. And consistently, what we observed is that there was a significant increase in IL-5 levels when we treated the mice with immune checkpoint blockade. And based on literature, for instance, airway injury literature or patients with allergy, it's known that IL-5 is a key cytokine involved in eosinophil differentiation, proliferation, and activation. So we hypothesized that this IL-5 may be important in our model. So we collaborated with junior group leader at the NKI, Daniele Thoma, she has set up a ex vivo patient tumor fragments platform, whereby she can treat ex vivo tumor fragments of patients with immune checkpoint blockade, and she has published before that this very strongly mimics the response or the non-response of patients. And well, we collaborate with her and show that also in her patient platform, IL-5 levels are increased when these tumors are exposed to immune checkpoint blockade, only in the responders and not in the non-responding patients. So we went back to the mice, and what we did is we neutralized IL-5 when we treated mice with immune checkpoint blockade. And what you can see, if we treat mice with anti-IL-5, this drastically reduces eosinophil levels in the bone marrow and also in the blood. And when we, on top of that, treat with immune checkpoint blockade, we do not, the immune checkpoint blockade does not overrule this effect, suggesting that IL-5 is needed also for the eosinophil increase when we treat with immune checkpoint blockade. And when we neutralize IL-5, also we mimic the eosinophil depletion. We see now that we lose the therapeutic benefits of immune checkpoint blockade and chemotherapy. So where's the IL-5 coming from? What is the source? Well, we know based on literature from airway inflammation, IL-5 can be produced both by innate and adaptive immune cells, in particular by mast cells, CD4 positive T cells, or ILCs. So to study whether in our setting the IL-5 is coming from adaptive or innate immune cells, we decided to make use of RAC knockout mice. So RAC knockout mice, they lack all adaptive immune cells. So we implanted memory tumors into wild-type mice or RAC knockouts. We treated them with immune checkpoint blockade. What we observed is that in the wild-type mice, again, the eosinophils go up in blood, but we do not see this happening in RAC knockout mice, suggesting that the adaptive immune cells are a source of IL-5. And when we analyze CD4 positive T cells, if we isolate them from mice being treated with immune checkpoint blockade, indeed IL-5 expression levels are higher. So what we did is we depleted CD4 positive T cells from our mice. And indeed what we see again is that when we treat with immune checkpoint blockade, now we don't see an increase in these eosinophils. So telling us that it are the CD4 positive T cells that are activated by the immune checkpoint blockade to make IL-5. So to validate this in patients, what we did is we went back to PBMC samples that we collected from a set of the breast cancer patients treated in the TONIC trial. We FACS sorted CD4 positive T cells from these samples, and we looked at IL-5 expression. And we see that in the majority of the tested patients, indeed, the immunotherapy treatment increases IL-5 mRNA levels in circulating CD4 positive T cells. So thus far, what I've showed you is that treatment with immune checkpoint blockade activates CD4 positive T cells that results in IL-5 production, and if IL-5 is increasing the eosinophil production in the bone marrow, which then leads to this systemic eosinophil accumulation. And these eosinophils are critical for having the activated CD8 positive T cells inside tumors so they can do their job and kill cancer cells. So what I haven't yet showed you then is how do the eosinophils enter the tumors? Because I showed you that in the mice, immune checkpoint blockade alone is sufficient to get this systemic accumulation of eosinophils. But in the mice, we need the chemotherapy to get the eosinophils inside tumors. So which mediator is involved in that? So what we did is, again, we performed a cytokine and chemokine array on plasma of treated mice, but now we also included the cisplatin treated groups. Again, what you can see is the IL-5 is coming up when we treat with immune checkpoint blockade alone. But if we add cisplatin to the treatment, we now see that also IL-33 is coming up as one of the key hits, which we validated also in the tumor lysates of our mouse models, that when we treat with cisplatin plus immune checkpoint blockade. IL-33, which is an alarmin, its levels are increased. So is IL-33 involved then in recruiting the eosinophils inside the tumors in the treatment groups? So here we collaborated with Rudy Bayard. He's at the VIB in Belgium, and he had developed an IL-33 trap molecule that we can use to treat mice with, and it captures away IL-33 from these mice. So we treated mice again with chemotherapy and immune checkpoint blockade in the presence or absence of this trap molecule. And what you can see in blood, we still see these eosinophils increased when we neutralize IL-33. And I think this is very much in line with that I just showed you, that IL-5 is the mediator that gets the eosinophils out of the bone marrow. But to get them inside tumors, as you can see here, we really need IL-33. Because if we capture away IL-33, we don't have this increase of eosinophils inside tumors. And we also now see that the tumor protective effect that we observe when we treat with cisplatin plus immune checkpoint blockade is lost when we neutralize IL-33. Because we have to inoculate this trap molecule daily, we were not able to follow the mice all the way to the end. But I think this shorter term experiment already shows that IL-33 is involved in this cascade. So what about IL-33 in patients? Can we find any indications there? So what we did is we went back to these sequential biopsies that we took during treatment, and what we see is in the responding patients, there's a positive association between the eosinophil signature in these metastases and IL-33 expression, which we do not see in the non-responders. Perhaps suggesting that also IL-33 in patients is important for to get these eosinophils inside tumors. So what I now showed you is this IL-5, IL-33 eosinophil cascade that is important for immune checkpoint blockade response in breast cancer. But of course, now the big question is can we use this knowledge to now improve immunotherapy response? And well, I think our mouse models are ideal models to test this, to provide proof of principle for this. And from the clinic really comes the question, is there a way that we can leave out the chemotherapy of this treatment effect? Because chemotherapy is a big burden for patients. So this is what we tried to do. And we took maybe a little bit of a simple approach to start with. But what we decided to do is to leave out the chemotherapy in our mice and to replace it with recombinant IL-33, with the hope that this will be sufficient to get the eosinophils inside the tumors to do their job. Indeed, what we see is that if we treat mice with recombinant IL-33, already alone it is sufficient to get more eosinophils inside tumors. But if we look at the activated T cell phenotype, we see that IL-33 alone does not activate T cells. It's really the combination of immune checkpoint blockade plus IL-33 that increases the activated T cells inside these tumors. And what we do see, and perhaps the effect is still relatively modest, but we also see a survival benefit when we now treat mice with immune checkpoint blockade plus recombinant IL-33. So indicating that there's at least a space here to improve immunotherapy by engaging this axis. In this experiment, what we did is we systemically injected recombinant IL-33. We have currently an experiment ongoing where we inject IL-33 intratumorally, because that may be more efficient. But yeah, to wrap this up, I think, well, for the lab, it was really very inspiring to 7 years ago to make this step, to invest into bridging the lab with the clinic. I think, combining these clinical trials and translational research with mechanistic research in preclinical models that mimic the patients, I think is a very powerful strategy to unravel novel mechanisms of immunotherapy response. I don't have to say this for this audience, of course, but I think, again, innate adaptive immune cell crosstalk is really essential to induce effective immunotherapy response. having shown this critical role for eosinophils in immunotherapy response, it really sets the stage for strategies aimed at engaging these cells to potentiate immunotherapy response. And we received funding from the Dutch Cancer Society to look for strategies to engage these cells to improve immunotherapy response. Well, there are many people involved in this. Started already many years ago with former Ph.D. student Kelly Kersten doing the first immunotherapy studies in our mouse models before immunotherapy became actually fashionable in the clinic. Many people involved, and I just show here the key players. Marleen Kok, amazing to work together with a clinical partner, Leni Voorwerk, Olga Blomberg, Hannah Garner, Lorenzo Spagnolo, but also, of course, the patients for their generous collaboration with samples that we were able to study, people that work in the clinic, and all the technicians that were involved in this. Really very much looking forward to hear any feedback from you, questions, suggestions. Thank you very much.
Thank you for an amazing talk. We have a few questions in the chat and also quite a few comments about what a wonderful talk this was. So thank you.
Thank you very much.
I'd like to ask a first question. This is beautiful work. Have you extended these observations to other kinds of solid tumors? Besides breast?
Yes, very good. Yeah, so in the clinic we have. So we have collaborated with Emil Voest. He's doing the drop study here. So we've looked at, and together with Miriam Salabi, we looked in her colorectal cancer patients, and we looked in non-small cell lung cancer patients. And in all of them except the MSI high tumors in the colorectal cancers, we see that the eosinophils increase is associated with response to immunotherapy. Mechanistically wise, with mouse models, we're now working together with Laila Akari at the Institute. She has glioblastoma and HCC mouse models. So with her, we are looking at eosinophils. And I've been talking a lot with many collaborators over the world, and people are now becoming interested in eosinophils. So I think that's good news. And if anybody of you have settings where you can study these, we are happy to share protocols to give you advice. They're not the easiest cells to work with, especially in vitro. It's a challenge, but I think there in vivo there are tools. We're now building together with Jacob van Reenen new transgenic mice to do in vivo imaging on these cells. So I think there's so much we still need to learn. So the more people work on them, I would say the better.
Great. Let me ask a couple of questions from the chat. Let's see. So Emilio Vero asked whether eosinophil accumulation is causative of response to immune checkpoint blockade. I mean, I think your results showed that.
In the mice, they're really causative because if we deplete them, we lose this therapeutic benefit. How they're causative, it's indirect, what we suggest, because it's via the activated T cells. We also have done a lot of in vitro studies to see whether in our setting they have direct cytotoxicity. In vitro, we can push them to directly kill cancer cells, but in vivo, we don't have concrete evidence for that.
Judy, I would like to follow up because I had the same question. Because Karin, yes, it's data within depletion is clear. However, the concept saying that mouse, I don't know by patient obviously, has to respond first, then produce IL-5, then eosinophil will be expanded, and then they come and start helping. So the question is basically, mouse should respond first, and then eosinophil will be engaged. So that's what the concept. That's probably that question. I share that concern. How to interpret the data?
Yeah, so I think the mouse has to respond, not necessarily. It's the CD4 triggering. So we see that CD4s are high in CTLA4 PD-1 expression. So I think also in the patients, we see that they are able to produce IL-5. So it needs to be kick-started via the adaptive immune system. I agree, but of course, it's all about, are we activating the whole cascade, right? Only activation of CD4 cells is not sufficient to kill tumors, and only activation of eosinophil, it's cells talking to each other. And if we target different elements in this circle, we break it. And I think that is what we see with all the myeloid cells. In the neutrophils, they also act often via other cells, right? And the macrophages. So, and I'm not telling that in every patient this is how it works, but we see that this cascade, yeah, can be very important. If we can engage it, it will be beneficial. But I see you're shaking your head. So what is your biggest doubt? Because I really would like to.
No. Because, well, for the neutrophil situation is slightly different. They are always there. They could be, there's an argument in help or not, but they are always there way before.
But the eosinophils are also
Right.
always there, right? We always
Right.
have eosinophils. It's just their
Your data suggests they really come after you treat them with a checkpoint. So that's what.
Yeah, but they
really
are already
the
there at very low levels, and they're
anyway.
really.
I mean, there's
Yeah.
a lot of questions. It's a fantastic story.
Thank you.
There will be question on cyclic F, but I couldn't resist to ask that. I'm pretty sure
Yes.
quite a few people ask you about it.
Yeah.
Because cyclic F has been implicated by quite a few papers, including 2 years ago,
Pitet with the neutrophils.
So
Yeah.
how are you sure that you deplete eosinophils but not neutrophil?
Yeah, that's a good question. So we also see a cyclic F on the subset of the neutrophils. Although again, here the MFI is much low. I didn't include the graph. I should have done it. The eosinophils are sky high. And then the alveolar macrophages and the neutrophils have a much lower MFI. So we did do a very careful analysis of the neutrophil populations. We do not see them altered. And I think the anti-L5 is a nice alternative. Some people use anti-L5 as an eosinophil depletion strategy. And we phenocopy our anti-cyclic F data with the anti-L5. And anti-L5, we don't see that the neutrophils are affected. So I think this really pinpoints to the eosinophils. We had the intention to build eosinophil deficient mice, but something went wrong with building the transgene. So we have to start over again. But I think there's a big need for more genetic tools, like for the neutrophil field as well, right? But I think having the anti-L5 and the anti-cyclic F, yeah, that helps.
We have a lot of questions. I think I'd like to ask people to raise their hands and ask in person. I think it'll be more effective. So first is Nan Zhang. Could you go ahead and ask your question?
Of course. Hi, it was beautiful work. So I have a question about spatial locations. I wonder if you're working on that. So, answering lots of questions, cellular interactions. Yeah. Have you done imaging, or, are you thinking about doing spatial transcriptomics, which obviously you probably are? And then can you comment on that? Thank you.
That's
beautiful
very
work.
much on our list. So we started it 2 years ago, a transgenic mouse, where behind the EPX promoter, we have Dendra, which is a photo-switchable molecule. And together with Jacob van Reenen, we were planning to then photo-switch eosinophils and follow them. What turned out 1.5 years later is that these mice, they almost don't have eosinophils. So somehow we disrupted the promoter working. So we went back to the drawing board. We're now making new mice. Meanwhile, we got a codex machine here at the Institute, so we're setting up, but it's very much geared towards human. And I have to say the biopsy, the sequential biopsies, they're so precious that we decided to use them for RNA sequencing. But for the mice, we would like to set it up. And we've done the simple immunohistochemistry. And it's in the paper, and I should have put it in my talk. We looked at the periphery, the outside of the tumor, the edges of the tumor, and inside the tumor. What we see with immune checkpoint blockade alone, already on the edges of the tumors, we see more eosinophils. But to get them inside the tumors, the memory tumors, that's we only see when we do the combination treatment. Suggesting that the location may matter, but we also know that in the treatment, they have a different activation status. So I think there's still a lot to learn there. What I didn't mention, but I think what may be even more important, we see the eosinophils very much increasing in the draining lymph nodes. And of course, that's also where the priming of T cells need to take place. So maybe I think we should be maybe focusing more also in the role on draining lymph nodes. So that is a little bit what we're trying to do now.
All right, thank you very much.
Yes, thank you for your question.
We have another question from MH.
Hi, Dr. D. Visser. So this is fascinating work. So I had basically one comment and another question. The comment is, did you basically look at the DBL-GATA1 knockout mice? That is basically eosinophil knockout mice available from Jackson, I think.
Yeah, regretfully, we work on the FEB background. And almost none of the mouse models are available on FEB. So that's why for us, we try to first do backcrossing, but making mice new is faster than backcrossing. So we are trying to make some of these again. Okay. So, yeah.
Yeah, and another question is your work actually nicely fit with the even T cell activating therapies, cytokines like say IL-2 treatment, and then you get the toxicity that is considered as like IL-5 dependent eosinophilia and eosinophil activation and toxicity. So in your model, did you see any toxicity or do you have any suggestion then, how to balance between, eosinophil mediated, T cell activation and then effector function versus, in T cell activating therapy, eosinophil mediated cytotoxicities, how to balance between these two, right? So if you want to recruit eosinophil and activate them in the tumor microenvironment, whether you will also activate the eosinophils in the periphery and get toxicity or not.
Well, that's very interesting. We don't see, well, I think the tumor is more harmful in the chemotherapy than we have not seen clearly overt eosinophil caused toxicity, but we now have a new animal pathologist here at the Institute. So I think it would be interesting to have a look at that. So yeah. At this stage, no, I don't know. Also, not really how to prevent that. To be honest, maybe if we have a better understanding of which type of eosinophils we need or which features of eosinophils are important, maybe then we can, fine-tune activation of this particular subset.
Great, thanks.
Thank you.
I just have a quick question based on what we've been discussing. In the patients that don't respond to immune checkpoint blockade, do you have any clues about which part of this axis is interrupted? Yeah, so that's something we really want to understand. And the IL-33 biology in patients is interesting because there are a lot of SNPs. There are certain SNPs in IL-33. So there's a circulating decoy IL-33 that we all have, but some people have way higher levels than others. So I would be very interested to see whether we can link this soluble ST2. So it's basically a soluble receptor for IL-33 that, because I've been talking to people in the allergy field and they advise me to have a look there because it may be that in the patients that have high levels of this soluble IL-33, that may be their limitation in this cascade. But yeah, so that is on my wish list to study in these patients. Yeah. Thank you. Seth.
Hi, Seth.
Well, hello. Nice to see you.
So nice to see you. Yes.
Did you check PD-1 on eosinophils? Do they express?
We did. We checked CTLA-4 and we don't see expression of that in our model. Because that was our first idea, that they would be directly activated by the immune checkpoint blockade, but we don't have indications for that.
Okay. You mentioned the lymph node as well.
Yeah.
Have you had a look at DCs? Do they also change with IL-5 increase? And
not yet, and I think we definitely should have a look there because it may be that the eosinophils may be talking to the disease as well, right? And that is this is the link between the eosinophils and the T cells. I think that's a very good, as always, Seth, a very good advice. And you are thanks to your research in the lab, we kind of started this whole adventure. Because,
Well,
you know,
yeah. Well, we did it together, right? We did it
well,
together.
yeah, you identifying the systemic inflammation really triggered us, right? And it's all these years ago that you said we need to look into patients. So thank you. You actually were very important in this story.
It was a team effort. So, but because I don't think that CXCL9 explains why the interferon gamma changes in the CD8s, because I don't think that the chemokine will regulate their phenotype in that way, right?
Well, so in the beginning, I was like, oh, this is a recruitment mediator. If you look
Yeah.
a bit in literature, there are also suggestions that it may be involved in their activation, but I'm careful about it. I don't know. But I like that idea of the, you know, we've more and more become interested in lymph nodes. I think the field is ignoring these too much. But, yeah.
Well, and so has the NKI done enough patients to know where there's a difference between males and females?
So that is, so for, of course, for breast cancer, we can't do that, but why do you ask that? Do you think there is?
Well, you know, there's all kinds of differences between, you know, like Th2 responses by gender.
Yeah.
So I'm just wondering whether if you go in, you know, if the eosinophil response is still the same, whether or not the response, you know, whether there'll be any differences between.
Yeah.
I don't know. Just
what we did do is we compared premenopausal and postmenopausal patients. And there we don't see an enrichment of pre or postmenopausal in this. But definitely interesting what you suggest.
Yeah. Okay, cool. So maybe something hormone related. Great to see you.
Yes, great to see you. Nice.
We have a question from Dr. Mitra.
Hi, Karin, it's a great talk, and just a quick question regarding the combination therapy like this, cisplatin versus checkpoint therapy. I mean, is it really driving in the bone marrow the T cells with a high, I think the PD-1s, I'm not sure exactly the PD-1 or TIM3 or LAG3, like a more exhaustive phenotypes of CD4 T cells? Or do you think just the systemic circulation when the antibody is given, it's just enough to drive IL-5 secretions by the CD4 T cells?
Yeah, an excellent question. So this is also something that people have asked me before, and we haven't yet been able to answer because it's basically shortage of people or too many questions we want to address. So what type of CD4 cells are actually doing this? I can't say that at this moment, but I think it's an important question. Where are the CD4 cells then located, the ones that respond? We pick them up in the circulation based on the tumor patient platform. They may also be inside tumors, but I think this is something we should be addressing. Yeah.
And another question I have, the IL-30, the recombinant IL-30 treatment to enhance the infiltration of neutrophils. So does it also modulate the TME? I mean, you know, of course, I can understand like it's an anti-tumor response by CD8s, but how about the total dynamics like macrophage or dendritic cells, like a global population-wise, how they are kind of skewing towards the
IL-33 is of course a very inflammatory molecule. So there's quite a bit. In the short time frame that we used it, we do see differences, not as dramatic as we were expecting, I have to say, but also systemically we are altering other cell populations. I was worried that we would be mobilizing more of these bad neutrophils. That's not what we observe, but we are now, there's a reason for that. We now move more towards intratumoral, because I think it's maybe better to really have it focused inside the tumor. And I'm not sure whether for patients IL-33, recombinant IL-33 is the way to go. I think we may want to have more sophisticated ways to engage these cells. But for that, our knowledge at this moment is just not enough to come up with alternatives. In the clinic, clinicians have actually proposed to maybe use parasites as a way. You know, there are parasites that are not so harmful for people, and we know parasites, they increase eosinophils. But I think now we don't have enough knowledge about whether the eosinophils that are increased by parasites, whether they're the same as you would be mobilizing with immune checkpoint blockade. So at this moment, I would not advocate to do that in patients, but maybe something to think about in the future.
Thank you.
Thank you.
So actually, we had a question in the chat related to what you just said, where I'm sorry, I can't pronounce the name, asked whether you thought that a disease of hyperactivation of the immune system like allergy or asthma could reprogram the innate immune system to protect against neoplastic disorders.
I think that is a very, very important question, but in these diseases of the many different immune cells, we know, for instance, neutrophils are also in these diseases activated and can be immunosuppressive. So I think it's intriguing to know. I think, especially in the context of parasites, there I can imagine that maybe because it's so much skewed towards eosinophils, that may be a way to maybe protect or improve immunotherapy response. I think at this stage it's too early to say that, but I would say that in all the years that we're studying chronic inflammation in the context of cancer, we learn from the pathways that people have identified in asthma and allergy and autoimmunity. It are the same molecules and the same cellular players that are involved in these diseases that we see back in cancer. So definitely there's a lot we can learn from that.
And I have a very quick question, technical question. In mice, aren't eosinophils Ly6G positive?
Oh, good question. Now I should know this. I know
Because
they have
so many
F480
of the biomarkers are very similar to resident macrophages.
Yeah,
so.
but if you use anti-Ly6G, we're not depleting the eosinophils. So if anything, I have to come back to that because I think my lab is now ashamed that I cannot answer this question, but I should be. I know they're low in F480, so we always. And you very nicely can pick them out based on the granularity with the SSC and the FSC and the flow cytometry. So they really stick out as a separate population.
Okay. Okay. So there's a question by Payal Yakota.
Yeah, I can ask if. Oh yeah, hi. Wonderful talk. Actually, you've kind of answered some of the questions that I had. Thanks to all the beautiful questions as well. One of the things I was wondering, and it's because you mentioned the parasitic infections, one of the things that triggers the eosinophil recruitment in parasitic infections is the epithelial, penetration, and that's what alarms the immune milieu and gets the IL-33 signaling going. So I was wondering, a couple of questions. One, do you think that there's something between the IL-33 and the eosinophil recruitment, and could we rule out ILC2s for the production of IL-5?
Yeah. I'm not sure about the ILC2s. My postdoc, he has done a big search into these cells, trying to get them out of the mice to study them. I'm not sure whether the REC knockout experiment is sufficient evidence, but regarding the IL-33, we've also, because we haven't really solved the source of IL-33, but we see IL-33 is pre-stored in cells and is then released upon stress, like you mentioned. And we do see that, especially in normal memory epithelial cells, when we do immunohistochemistry, they are quite intensely staining for IL-33. And sometimes in the tumors, we see that the staining is less. I'm not sure whether that means that it is released or not. So it's a little bit a challenge to really pinpoint where the IL-33 is coming from.
And then the other question was, it's impossible probably in the human studies, but in the mouse model, could you perhaps determine the source of eosinophils? Are they tissue derived, or are they coming from the vasculature perhaps?
Yeah, I think in the end they come from the bone marrow.
Okay.
But that's where they are produced, and where we really see, well, at least we see that they are increased when we treat with immune checkpoint blockade in the bone marrow already. And some of the progenitor cell, I think there's still, it's not really clear exactly which progenitor cells and how, but we see that the early eosinophils are also increased. And that fits with this IL-5 link, that it is involved in the differentiation of the eosinophils in the bone marrow. So thank you.
Thank you so much.
Thank you. It's wonderful discussion. It's really nice.
Okay, I think we have time for one more question by Gayatri.
Gayatri, thank you. Wonderful talk to Dr. Devas. I wondered if you are able to elaborate a bit in the context of colorectal cancer. You mentioned in passing your work there. Role of eosinophils, particularly with the involvement of the gamma delta T cells and the epithelial surface there. If you don't mind, I know the focus is TNBC, which is amazing, but if you could please elaborate, thank you.
Yes, so to be honest, there's not so much I can say there. We've looked with Miriam Salabi, especially in the high mutational load colorectal cancer, what we there in the blood see, because there we did not see this, well, they all respond, basically, these patients, but we don't see that eosinophils went up, but the baseline levels of eosinophils in these patients seemed higher than in the other cancer patient cohorts that we looked at. But I think we need to have a better look there. It may be that there is already a kind of well, maybe a little bit of a smoldering immune response that causes these eosinophils to be higher. But the link with the gamma delta T cells also there, yeah, wonderful question, and I cannot answer it regretfully. But
I look forward to data from your group soon then. Thank you.
Thank you.
So not to add yet another dimension to your research, but I wonder if you've been thinking about the role of the microbiome in
thinking of that, but not studying it at this moment. It's mainly because we don't have the expertise, also not in-house here, but definitely a very important element. And
I know it adds another layer to all of what we do.
Yeah, and it is. For us, it's already handful to work on this genetic makeup and how the genetic makeup shapes the immune landscape. It is a little bit scary how diverse and how complex the regulation of the immune system is in a cancer context. But I'm sure, and based on literature, we're slowly getting there. Deeper understanding, because we really need to move more towards this personalized immune intervention studies. So the more knowledge we gather, the closer we get.
Well, this has been a wonderful session and really insightful and original work. So thank you so much for presenting today.
Thank you. And I really love the discussion. It's really I got a lot of new ideas. So thank you very much for hosting me.
All right. So have a good evening.
Thank you.
And everybody else a good day.
Yes.
And we'll see you next month at our retreat. Send in your abstracts, please.